Just keep pressing buttons and something happens. Well, thank you all for joining us today. It's a pleasure to introduce Dr. Art Hunt to you. Before we get started, we're going to try more of our experiment. Is there anyone new in the audience for introductions today? I see lots of faces I don't know, but that may mean you're not new. Anybody want an introduction? No. Okay. So here we go. Art has been with us for 33 years. He earned his bachelor's degree in biological sciences from the University of Lowell, which is now called the University of Massachusetts Lowell. But I'm probably not saying it right.
That's close enough.
Okay. And a PhD in biochemistry from Brandeis University in 1982. He did a postdoc at the Rockefeller University in the lab of plant molecular biology. And he joined us when we were called Department of Agronomy in 1985. He is a full professor in our department and has been for some time. And the two big honors that he's received lately have been, he was the university research professor in 2016, and then he became an American Association for the Advancement of Science, or AAAS fellow, in 2017. So, without further ado, I believe he has a test for us that I have been failing miserably, so I'll hand it over to you, Art.
Okay, give me a second here to get wired up. Okay, well, thanks for the introduction, and thanks for the opportunity to everybody for showing up and listening to different things that are going on in my laboratory. So I start off with this slide here. So this is a test that unfortunately just reminds me of how old I am. So, and so the bottom slide, the bottom part of this is from a movie that's 50 years old this year. But really the take-home message for this is turn off your cell phones. Okay, so I'll give everyone a second. And then move on to the talk. So I came up with a real clever title for the talk, and you'll see why this title is clever as we move on here. Polyadenylation in agriculture. Maybe it was a trick just to get a lot of people here to listen to some of the things that are going on in the laboratory. So this is the 60-second version in case there are refreshments waiting out for us here. So we're going to talk a little bit about why you should be interested in polyadenylation, why it's the most important thing in the world to be interested in. Then the main part of the talk will go over some things that have been developing in my lab probably for 10 years running now, studying the connections between polyadenylation and stress. I say abiotic stress here, but actually there's one project we have going on that studies the interaction between polyadenylation and plant pathogens as well. And finally, I'm going to introduce you to a kind of a side project that's got me really excited and has nothing to do with plants. But I think it's a very interesting side project that's going to go lots of places. So because sometimes the slide gets lost at the very end, I think it's important at the beginning to acknowledge everyone who's been involved in the work that I'm going to be talking about. Members of my laboratory listed here, other labs in the College of Agriculture at the University of Kentucky, and collaborators at UC Riverside, Colorado State University, and Kentucky State University. And I want to emphasize the collaboration with people at Kentucky State. So this is a collaboration I've had with a faculty member in the Department of Computer Science. And so I've had some very good and helpful students writing some of the simple computer programs for the analyses that we do. So why polyadenylation? Why should everyone get up and jump and cheer about polyadenylation? So putting things in a little bit of an agricultural perspective, and this is kind of the best context that I could bring out here. So everyone knows that many factors contribute to growth in agricultural productivity and so on and so forth. This is true for all plants. So developmental factors, antibiotic stress, biotic stress. So we can put this in fact, in my mind, I put this all very much in a gene-centric view where everything feeds into gene expression. So these different factors feed into transcription, translation, and so on and so forth. So in this context, of course, polyadenylation is very important. So polyadenylation sits right at the heart of messenger RNA biogenesis. Polyadenylation is a process whereby poly A tail is added to a messenger RNA. And of course, we all know that the poly A tail is extremely important for the functioning of the messenger RNA. Without a poly A tail, the messenger RNA is not translated by ribosomes, and so you get no protein, and so on and so forth. The poly A tail is added to the messenger RNA in a post-transcriptional RNA processing reaction. It's summarized in this slide here. So there are some salient points here. The important, really the thing that you should take home from this, never ever forget, especially if you talk to me about this anytime, is that this is an RNA processing reaction. The poly A tail is not coded for in the DNA. It's added after the RNA is made, processed by an enzyme called poly A polymerase, curiously enough. Well, it turns out that in most eukaryotes, most messenger RNAs have more than one potential poly A site. So this reaction is presented pretty simplistically, and I drew this to emphasize that there are specific RNA sequences which tell the apparatus in the cell that this is a substrate for processing and polyadenylation. Most messenger RNAs have more than one such signal in them. And this means that most messenger RNAs can have different fates. So messenger RNAs can have fates whereby they're processed and polyadenylated to give a messenger RNA that will translate into the protein that you're interested in studying. But there are many other forms and fates of the messenger RNA that are also made and are also important for regulation. In this seminar, I'm going to be calling these non-canonical isoforms. These are just messenger RNA isoforms that might be translated or might have altered protein coding capacity. They might have altered functionality, stability, translatability, or so on and so forth. So these are isoforms that really do something other than give you a normal functioning protein. Well, for 10 years or so running in the general field of molecular biology, alternative polyadenylation has been something of a rage. And it turns out that in mammals and humans, in medical sciences, alternative polyadenylation is extremely important. It's been shown to be crucial for oncogenesis, for cancer development. It's crucial for in stem cell biology for different diseases. And so I mentioned preeclampsia because this is something that's brought to my mind a year ago at a meeting, and I thought the story was very, very interesting and exciting. And also just for general cellular proliferation. So it really impacts everybody, whether you know it or care about it or not. It also has been known for many, many years to impact plant growth and development. And so over the years, there have been reports of alternative polyadenylation impacting genes which are involved with self-incompatibility, amino acid metabolism, more interestingly, flowering, defense responses, and so on and so forth. So many of you probably are familiar with one or more of these examples, and more generally, you're inclined to think about alternative polyadenylation and these effects as affecting important single genes, your favorite gene, the gene your thesis is being written around, and so on and so forth. Well, for this talk, I'd like you to kind of forget or put this tendency aside. And so to do so, I'd like you to look at the flashy thingy so that you can start with a clean slate, okay? And we need the clean slate to better appreciate what I'm going to describe in the next few slides. So I'm going to talk about polyadenylation and stress. So again, I apologize for the typo here. It's just stress in general. Studying how polyadenylation interacts with and contributes to stress responses in plants. The reason in my laboratory for going down this road came out of some studies which were published 6 years ago and were another 2 or 3 years in the making. So we got interested in studying poly A site choice genome-wide and came up with the idea to characterize things, group poly A sites in terms of canonical versus non-canonical. And we noticed a very interesting association of non-canonical polyadenylation sites with genes that have been annotated as being associated with different stress responses, biotic and abiotic stress responses. And so these results are summarized in this slide here. So this really is just a summary of what we call a gene ontology analysis. We just collect genes which are associated with non-canonical polyadenylation and ask if there are any functional definitions or whatever in the set of genes that are overrepresented when you compare the set of genes with all the genes, in this case in Arabidopsis. And indeed, there seems to be an association. This is just an association. This doesn't tell us a lot more than that, but it does give us incentive or impetus to ask the question, is the production of non-canonical polyadenylated RNAs actually associated with stress responses in plants? So we set out to do this, and we took what is the workhorse, the experimental approach in the laboratory to this, and almost everything I'm going to talk about for the next 30 minutes or so at least is built around this experimental approach. We use this because I developed it in my lab with my own hands, and I make everyone else learn the approach. And it's been very useful and taken us a lot of places. So I'm not going to get lost in all the details in the next few slides, but I just, there's a three-tiered approach here. The first happens at the bench. We go and take RNA and convert RNA into short cDNA tags that we sequence in very, very large numbers. We take these sequences to the computer and we do all kinds of analyses with these sequences. We place them on reference genomes. We characterize them in various and sundry ways to get information such as gene expression, tag distribution, poly A site usage, and other analyses. So 15 minutes in, it's time for a commercial. I thought it would be useful just to point out again. So most of these techniques have been developed in my laboratory, and one thing I found is that they're very useful for many experiments and uses other than just studying polyadenylation in Arabidopsis. So the approach that we take, illustrated here, I call PATSI. It's a rapid, very low cost method for getting not just poly A site information, but gene expression information in all organisms. It reduces the cost of next generation high throughput sequencing by more than 90%. And it's adaptable again to any organism. I've taken this method and technology on the road, and so it's been taught to laboratories coast to coast and around the world. More than 50 laboratories, more than 2,000 students, undergraduates impacted by this. And I'm always interested, as you will see in all the collaborations I develop, in working with other people to help cut down their sequencing costs and use this in their own research. So people might wonder, well, why should I be doing poly tags? Well, the technique actually can be adapted and has been adapted to do more traditional RNA-seq. That's right, this is another commercial. You never just get one commercial in a commercial break anymore, okay? And so RNA-seq has also been used in workshops and is being used at universities and undergraduate focused institutions worldwide. We actually use this in ABT 495, and we have been for several years, and I think it's been very successful. So the third commercial in this commercial break, this is something that I mentioned at our departmental faculty meeting a few weeks ago, but I think it's something that all trainees and members of the department should be aware of, and that is we have a bioinformatics set up in the third floor of this building that's very user-friendly. It's been established with very generous support from our department. 2 computers, lots of very useful software, and the capability to adapt to your own particular research. And so this is something, again, we've been using with ABT 495 for several years, and it's something that is available for use for trainees in the department. So what do we use this bioinformatics capability for? Well, we take all of these RNA-seq tags, so what we do is we convert populations of RNAs into short tags. If you paid attention to the method, you'll see that every cDNA that we make queries the junction between the messenger RNA and the poly A tail. So when things work right, every sequence maps the very end of the messenger RNA. So we take these sequences, demultiplex them, et cetera, and so on, and place them on a genome, and we get an output which can be illustrated something like this. So in this slide, every green tag is an individual sequence. We usually will map between 10 and 100 million individual sequences in an experiment, and then we break this up into individual genes. And so the way this picture works is this is a representation and annotation of a single Arabidopsis gene, and you can see that we've got about 100 tags that map to different places in the gene, and each file here represents a different polyadenylation site. So we take this information and we calculate a metric for every site, for every condition. So the metric is illustrated here. So basically what we do is we calculate the fraction of all the tags in this sample which map to A, to B, to C, or D. So A, B, C, and D each get a different metric. And then we compare these metrics between treated and control plants, generate a very simple ratio, transform it, and this is what we use in further analyses. Now, we're not doing this on a single gene basis. What I'm interested in and where we're going with this is to study classes of polyadenylation sites, canonical and non-canonical. And the thing we're interested in is asking, is there a connection between non-canonical poly A site usage and stress? So what we do is we sort the different sites. So each of these sites would be in a different class, 5' UTR, introns, CDS, and 3' UTR. These would be canonical poly A sites. We sort things like this, and then for each class, we take this metric, we pool them, and we analyze them in a very simple way. We just go and represent the aggregate values for each class and compare things using very simple plots, box plots. Box plots that also have the nice feature that you can apply statistical tools, notches in this case, and get a very quick graphical representation for things which might be significantly different. So we take this approach and we study plants which have been subjected to different stresses. So Guojie Hao, in her thesis project, studied an osmotic stress situation. So she subjected Arabidopsis plants to high concentrations of mannitol. This approximates drought stress. And then ran through the process and asked the question, do different classes of polyacites or do different isoforms that are derived from classes of polyacites change in the course of exposure to drought stress? So the box plots shown here, and I just put arrows here just to show you what's of interest and significance for this experiment. And what we see here is that indeed the relative abundance of isoforms, messenger RNA isoforms derived from polyadenylation within coding regions and within 5' untranslated regions, in fact, go up quite a bit in drought-treated plants. We do the same experiment under conditions which approximate hypoxia. So Laura Di Lorenzo at my laboratory, in collaboration with Julia Bailey-Saris and Reed Sorensen at University of California, Riverside, did this experiment, obtained these results, similar to what we saw with drought-treated plants, non-canonical isoforms, which correspond to polyadenylation within coding regions and 5' UTRs. Their relative levels or abundances increased upon exposure to hypoxia. Interestingly enough, there's something a little different. Intronic isoforms also seem to increase. So Laura, again in collaboration with Pradeep Katru upstairs in the Department of Plant Pathology, has been conducting a fairly extensive set of experiments with pathogen-treated plants. A representative outcome is illustrated here. So this is just Arabidopsis treated with a virulent clone of a bacterial pathogen. And again, Laura sees that messenger RNA isoforms, which correspond to the polyadenylation within protein coding regions and 5' UTRs, are their relative abundances are much greater in the pathogen-treated plants than the control plants. So in my little world, agriculture and plant science is all about Arabidopsis, but I'm not so naive to realize that, well, there's probably a little bit more to life than Arabidopsis. So Manohar Chakrabarti, Laura, and collaborators at Colorado State University set up a fairly large experiment studying stressed sorghum plants. So the results of this study are summarized in this slide here. So this is this is kind of a busy slide. So what I've done is I've just boxed off the results according to the class of non-canonical or canonical polyase site, and presented the results obtained with drought-stressed, salt-stressed, or heat-stressed sorghum plants. And so I put a purple line across here just to indicate that this is where the medians for these different boxes would lie if there was no change in polyacite usage, the usage of these different classes of sites in aggregate. And I think it's pretty easy to see that for all three stresses, drought, heat, or salt, non-canonical messenger RNA isoforms, the relative abundances do in fact increase by quite a bit. In fact, something that's a little bit different in sorghum compared to what we saw in Arabidopsis is the redirection of transcriptional output into these isoforms is so great that the usage of canonical, or the abundances of canonical messenger RNA isoforms actually decreases noticeably and measurably. So to sum things up, what we've seen in these studies is the relative abundance of isoforms which are derived from messenger RNA polyadenylation within protein coding regions and within 5' UTRs seem to be uniformly impacted by a wide battery of stresses. And they're impacted in ways such that the abundance of these isoforms increase. Isoforms with 3' ends, which end within annotated introns, behave a little bit differently. So in some stresses in Arabidopsis, their relative abundances increase. With other stress treatments, they don't. In sorghum, we see that there's a uniform, modest increase in the abundances of these respective messenger RNA isoforms. So interestingly and importantly, we see similar analogous results in Arabidopsis and sorghum. So it's conserved in all plants, okay? Not necessarily, but I think there's a commonality which suggests that what we're seeing is probably a general feature of the response of polyA site choice to stresses. So what might all this be telling us? So I think this is where things get interesting to me, and this is what suggests new approaches to take in the laboratory in this project. So we need to go back and recall that non-canonical messenger RNA isoforms are, for the most part, non-functional. They won't give you a functional polypeptide. And in fact, other research in the laboratory has shown that most of these isoforms, excuse me, in addition to not giving or yielding a functional polypeptide, they're less stable than canonical messenger RNA isoforms. So this is, so these in aggregate are isoforms which seem to redirect transcriptional output into non-productive pathways. And the main conclusion from this work is that plants seem to redirect a substantial part of their transcriptional output to the production of non-functional messenger RNA. So there's a typo there. So it should be non-functional messenger RNA isoforms when they're treated with stress. So plants respond to abiotic and biotic stresses in a number of global general ways. Many mechanisms, so the aim, I think, for all these mechanisms is to redirect transcriptional output and translational output away from most proteins into a subset of proteins. So translational output, protein synthesis, is reprogrammed by stress in plants, also in animals and yeast. The mechanisms that underlie this redirecting of transcriptional and translational output are many and varied. There's a great deal of transcriptional and epigenetic control, which turns on and off large sets of genes. There's translational control. I think translational control is very, very important for the responses of plants, animals, yeast, and so on and so forth to stress. Messenger RNA metabolism also is very important for redirecting this global change in gene expression. What we've seen with Arabidopsis and sorghum suggests that alternative polyadenylation is one of these global mechanisms which redirects transcriptional and gene expression output into non-productive pathways and therefore contributes to the general change in gene expression output in response to stresses. So this can be illustrated as I've shown here. So in a normal situation, you get protein production, stress redirects a lot of the transcriptional output away from translation and into storage granules, stress granules, RNA turnover, and so on and so forth. And alternative polyadenylation is one of the general global mechanisms that helps to redirect transcriptional output away from protein biosynthesis. Yes.
So should the alternative RNA forms, could they have regulatory functions in themselves?
You didn't look at the flashy thingy, did you? So the answer is yes. And so one can go and look at individual isoforms and hypothesize different regulatory functions for each and every one of them, certainly. And so not so much in my lab, but in my collaborators' laboratories in China, there's a great deal of interest in looking at different individual isoforms and correlating their production or disappearance with microRNA, small RNA production, targeting, etc., and so on.
I didn't look.
Huh?
I didn't look at it.
So where are we going with this project? So this is kind of where we're at now. So some of the things that we're looking at currently, some projects that are ongoing, some that are going to be set up. We're interested in studying mutant plants which are affected in different polyadenylation factor subunits to start to ask questions about the connection between stress and polyA site choice. How does stress actually change the place in the messenger RNA where processing and polyadenylation occurs? We're also studying mutants affected in RNA quality control. So something I've mentioned very briefly, but it's a very big part of this project. It has to do with the connection between non-canonical RNA isoforms and RNA quality control. And realizing this, it's important to be studying mutants in Arabidopsis mainly that are affected in various of the RNA quality control processes. By RNA quality control, I mean the processes whereby RNAs are broken down or turned over in a regulated fashion. And finally, we continue to do biochemistry on individual polyadenylation complex subunits, again, seeking to make biochemical connections between stresses and polyA site choice. So we're also interested in exploring and getting deeper into the possible contribution that APA might make to survivability under stress. And so I also didn't look at the flashy thingy, and so we inevitably have to go and look at individual genes to ask, to get insight into these questions. But something that we're also interested in doing, so my lab and collaborators in China are trying to come up with different screens to identify mutants which might be impaired in stress-induced alternative polyadenylation. So now for something completely different. The reason that my title was very simple was because I couldn't figure out a way to squeeze in global alternative polyadenylation, stresses in plants, and the targeting of the poly-A complex in parasites which infect livestock. That would be a pretty gruesome title. So, but livestock and plants are both agriculture, hence the title. So I was pretty excited about this project for a lot of reasons. So I've been talking to the trainees in my laboratory about this project more than 20 years ago. And the first person I talked to about this, when I told him what I was doing, he said, "Yeah, I remember, you're actually going to do this?" And so the rationale behind this project lies in a couple of papers, which I'm sure everyone read and noticed, in EMBO Journal and in Nature. It was reported a little bit more, about a year and a half ago, that a compound that I'm going to call AN3661 because the company that made it calls it this, is a very effective inhibitor of the parasites that cause malaria and toxoplasmosis. Well, there's a lot of these compounds floating around in the literature and in the scientific and pharmacological community. But what's really interesting about this compound is it's been shown that this compound targets a highly conserved subunit of the polyadenylation complex, a subunit that I call here CPSS73. So this is very interesting. It's important because not only does it target CPSS73 in these organisms, it has no effect on the growth of the hosts of these organisms, which in these studies were cultured mouse or human cells, or even live mice. So this raised a question in my mind, and again, this is a question that I've been pondering for a long time, and this is there something particular or interesting or different about polyadenylation in this phylum of organisms? So a little bit of a review. Polyadenylation is catalyzed by a complex. So this is a picture of the complex, and if you really squint, you can see the names of the subunits in here. But I made it easier for you to find these names. And so it's a fairly sizable complex. I won't get lost in all the names. I just want to point out that the enzyme that adds the poly-A tail, poly-A polymerase, I highlight in red. The target of this inhibitor, CPSS73, is actually the endonuclease, which processes the RNA. There are two other subunits that I highlight in black here. These are the subunits which, in the past 3 years or so, have been shown to actually bind and recognize the primary sequence determinant for polyadenylation in an RNA. So this complex will only cleave and polyadenylate an RNA that has a polyadenylation signal, represented here by A2UA3. Well, all this is a nice exercise to think about on paper, but it turns out that there are many more organisms in this phylum that include Plasmodium and Toxoplasma, including a couple of parasites that Dan Howe across the street works on. And so, actually inspired by an undergraduate in my laboratory, we set out to do a small collaboration to ask some simple questions about polyadenylation in these two parasites. So these are apicomplexans, which cause disease in horses, livestock, dogs, and so on and so forth. So Ashley Stevens in my laboratory set out to ask two questions. Actually, first she set out to ask one question. So the original goal for this project was to identify the apicomplexan polyadenylation subunits then do sequence alignments and get some information that you could use to do some biochemistry, 2 hybrid assays, something like this. So the way you go about this is you take genomes which have been sequenced and annotated, and you search these genomes with using as bait very well characterized subunits of, in this case, the human polyadenylation complex. You could do this with a plant or a yeast database as well, but actually did this with the human complex. So the results of the blast search are summarized here, and so I've gone and taken the raw outputs and put it into a form that's easy to read. So basically, the height of the bar here indicates whether or not there is an ortholog of these different subunits in the genomes that Ashley looked through here. So these are just 3 apicomplex genomes, and these are probably the 3 that have been the best characterized, and they're fairly complete. And what's remarkable, what Ashley's found, was that when you go and look, you can only find 1 of the CPSS subunits and very few of anyone else in this list in these genomes. You can look at the rest of the subunits and realize the only thing that really jumps out and seems to be there is poly-A polymerase. So Ashley's blast results can be summarized here. So I'll acknowledge it's important to look at this critically. So we're just making a leap based on the fact that we can't find things in genome databases. Okay, and it's always possible that databases are not complete, they're not well annotated, they're garbled or scrambled or so on and so forth. But the fact that we're missing 85% of the complex, it does not agree with the fact that these genomes, especially the toxoplasma and plasmodium genome, are not 85% incomplete or garbled or whatever. So there's a disconnect here, a disconnect that we're interested in studying. And the disconnect, I think, is very interesting because among the things we can't find are the 2 proteins which bind ATUA3 in animals and probably in yeast and plants. So Ashley's project became one of, instead of finding proteins and doing expression and biochemistry, 2 hybrid interactions, to one of asking, if these proteins are missing, what about this poly-A signal? Is this poly-A signal there or not? So in order to do this, we pulled out the PAT-seq library approach, made a lot of libraries, sequenced them, analyzed them, and so on and so forth. The analysis is a little different from what I showed you before. Basically, we use this information to identify poly-A sites genome-wide. Then we take every poly-A site, line them up, and analyze the sequences upstream and downstream from the actual cleavage site. The analysis is very simple. For, in this case, 6,000 sites, we ask at each position, what is the percentage, percent composition for each of the 4 bases that are in an RNA? Okay, and the way this works, so this is actually results that were published by Bin Tian a long time ago. This is an analysis of human poly-A sites, and you can see it's a little bit compressed, but there's a decided peak of A's here. This represents the ATUA3 polyadenylation signal. So this analysis is a fairly straightforward way to identify at least hints of specific signals which are associated with cleavage and polyadenylation. It pulls out ATUA3. In plants, it pulls out the A-rich near upstream element, which is probably functionally analogous to ATUA3. It also pulls out the cleavage element and so on and so forth. Well, when Ashley did this with Sarcociscus, she's also done this with Neospora and Toxoplasma. lo and behold, a very very obvious peak of A's at the position which corresponds to ATUA3 is in fact apparent in the poly-A signals for these apical organisms. So we conclude from this a couple of things. One, we have a hard time finding orthologs of most of the polyadenylation complex subunits in this phylum of organisms. But we see what is probably an analogous polyadenylation signal in these organisms. So this brings me to the end of the talk, the undiscovered country, where we're going with this project here, slowly but surely in collaboration with Dan Howe. We're interested in asking, where are the missing subunits of the complex? And we're interested in this because these are potential drug targets, something which is involved in a fundamental basic process, which is very different in pathogens of livestock and humans, is a potential drug target. And the precedent set by AN 3661 means that, well, this is a viable route to go for finding drug targets. We're asking questions about how alternative polyadenylation may or may not impact pathogenesis as well as growth and development in organisms in this phylum. And finally, we through a very circuitous process, got our hands on some AN 3661, and we're studying the interaction between this drug and the organisms, the parasites that Dan studies, to ask how this compound affects gene expression in these organisms. And that's the end of the road for now for this project. I'm delighted to take questions. Peter.
I wonder whether the alternative polyadenylation is done by the same complex as the canonical one.
That's an excellent question. And so the jury is still out on this. So this is one reason that we're studying poly-A factor mutants, because the prediction is that a mutant in the complex, if the complex is involved in the usage of these sites, it will actually affect usage of these sites. So I would say that, so we studied 2 or 3 mutants. One of them, there seems to be an effect, but it's not on all classes of sites. But we don't have answers that I consider to be satisfactory. So this is one of the big reasons we're studying mutants, is to get a good handle on this question. Jason.
So when we expose organisms to toxicant stress, it seems like in sort of the initial inflammation response, the mRNA levels change in many different genes, and things sort of settle down. And it seems like kind of like a short-term response that involves a lot of pathways, and a long-term response with fewer. So I was wondering, do you see anything with the intensity and duration of stress with choice of alternative?
Yeah,
and
so
that might correlate somehow with that.
Right. So yeah, so our experiments haven't been done with that kind of resolution. So generally we do 1 or just a few treatment time points and so on and so forth. And really, just because of the way we do the experiments, it's pretty hard to go and to do things 15, 30, 60 minutes after and get a satisfactory answer. So that's a really good question. So in the plant systems we studied, we don't have an answer. Hung in.
So you said the production of non-canonical mRNA is conserved between autologous genes from different species.
Well, so the effects of non-canonical isoforms, the effects of stress on the production of these isoforms as groups is conserved. When it comes to individual sites, not so much. So we've done this comparing Medicago truncatula and Arabidopsis. There's not 100% conservation. There's a lot of variability in non-canonical alternative polyA sites, gene by gene in these two organisms. But there is some conservation. So there's a lot of conservation in sites which lie within protein coding regions. Not so much so with sites that lie within introns. So
is alternative polyA site predictable?
no. In other words, is there a computer program that you can use to say that's going to change? No.
Another question. So we know there's a association between production of non-canonical mRNA and stress.
Right.
So how can we use this knowledge
How?
to enhance tolerance?
That's a great question. So I guess the idea is that part of the way the plant survives is to redirect output. And so it makes sense that, well, perhaps if you could tweak things to improve the efficiency of this, then maybe a plant would survive stress a little bit better. That's the idea. So that's a pretty challenging project because the scope of what I've described here is pretty wide-ranging. And it's hard for me to imagine a way to tweak this that won't have pleiotropic effects that have nothing to do with stress. So this is a tough question. The first thing I want to do is to find mutants that are impaired in the production of non-canonical RNA isoforms and find out what the consequences are. And so that's one direction we're kind of pointing towards.
Did you look at different developmental stages? Did you say 2 time points? Was they different stages? Or?
No, we haven't done a lot of developmental biology work in my laboratory. So I have a very active collaborator in Xiamen University in China who has done an extensive developmental series in rice. And they see a lot of stage-specific polyA site usage in rice.
One more question. Just curious about the mechanism behind alternative polyadenylation. Epigenetic?
Pardon?
Can be genetic mechanism behind alternative polyadenylation?
So the question is, are there genetic or epigenetic mechanisms behind determining polyA site choice? There's some evidence which connects epigenetic modifications with polyA site usage in specific genes in Arabidopsis. So this is an attractive mechanism. I'm not sure what you mean by genetic mechanism. Just mutations in polyA factor 17. So we know that. So with one of our favorite gene codes for a protein called CPSF30, and we know that when you take CPSF30 out of plants, you get a global change. You get large numbers of new polyA sites which are created.
I was just wondering if the host or the parasites you were studying can live free of the host. Reliving. I mean, it's not.
Well,
like
so most of these have multiple hosts.
Right.
And
I'm just trying to remember.
They're not free living. I don't think Dan Howe grows these in a petri dish. No, he grows these in cells.
Could they borrow parts of polyA? part of the cleavage complex?
Well, so I've actually, I had reason to think about this possibility, but Dan kind of shot me down totally with that. So I don't know enough about cell biology of these. So we got very preliminary 2 hybrid screen results for the toxoplasma CPSF73, something which suggested something like this. And Dan said, nah, this is just an artifact. So, Hanyan.
So when we talk about alternative splicing, usually we say more than 60% of the gene undergo alternative splicing. So for production of non-canonical mRNA, how often this happens?
That's a good question. I should have the number off the top of my head. The number of genes that are impacted by non-canonical polyadenylation. Manahar, do you remember?
It should be in the same range, if not more.
Yeah.
It's
So
in the same range.
So this is just non-canonical APA. So I'm guessing maybe 50%. Michael.
What's the stability of the non-canonical poly-A transcript versus the cognate canonical?
So it depends on the classes.
Yeah.
So I think the ones that you can predict to be less stable are RNAs that are polyadenylated within protein coding regions are going to be by definition non-stop RNAs. They have no stop codon. And these have been shown in other systems to be unstable. They're targeted while they're on the ribosome for degradation. And so we know that this class of RNAs in Arabidopsis is decidedly less stable in aggregate than canonical RNA isoforms. Intronic RNA isoforms also are somewhat less stable. 5' UTR derived isoforms, I don't think their stabilities, their stabilities aren't that different from canonical RNA isoforms. And I mean, they're kind of bizarre. I don't understand that at all. David.
Is there an advantage to post-translational gene shutdown versus transcriptional? Is it faster? Is there any advantage to stopping the functional mRNA rather than just not making it?
There probably are many advantages, but out of my mouth, this would be largely speculation or just what people talk about at meetings and so on and so forth. So post-transcriptional, post-translational processes, they help to clear things out faster. So if you need a new set of proteins, you need to get rid of the capability to synthesize or store or have the old proteins. So removing proteins in RNA is important for that. On top of that, obviously, there's a lot of transcriptional output that is going towards the production of non-productive isoforms in the stress plant. And this probably means that for whatever reasons, transcriptional shutdown or downregulation either is not efficient or is not very useful or helpful for responses. So it is possible that the transcriptional machinery needs to be engaged in some way to recover from the stress when it goes away.
So it can provide more fine-tuning.
Yeah, so that's the exact phrase that we use in most of our papers. So this is a means to fine-tune gene expression.
We have one more question for Art.